Hello guys, welcome back to my YouTube channel. I am Karina and our today's topic is of classification of enzymes. To start with, what is enzymes? Enzymes are biological catalysts that accelerate chemical reactions in living organisms. They are typically proteins, although some RNA molecules also exhibit catalytic activities. Enzymes are crucial for various physiological processes, including digestion, metabolism, RNA replication, DNA replication, and cellular respiration. They alter or speeds up the chemical reactions that occur in the cell. Key characteristics of enzymes. The first one is catalytic function. Enzymes increase the rate of chemical reactions without being consumed in the process. They lower the activation energy required for reactions, enabling them to proceed more quickly and efficiently. Specificity. Enzymes are highly specific for their substrates, the molecules they act upon. This specificity is due to the unique 3D structure of the enzyme, particularly the active site where the substrate binds. Active site. The active site is the region of the enzyme where the substrate binding occurs. It is typically a pocket or groove on the enzyme's surface shaped to fit the substrate precisely. The interaction between the enzyme and the substrate at the active site leads to the formation of an enzyme-substrate complex. Mechanism of action. Enzymes facilitate chemical reactions by various mechanisms, including orienting substrates, which is correctly to encourage a reaction, inducing strain on substrate bonds, making them easier to break, providing a favorable microenvironment for the reaction, forming temporary covalent bonds with substrates during the reaction process. Cofactors and coenzymes. Some enzymes require additional non-protein molecules called cofactors, which can be metal ions or organic molecules called coenzymes, often derived from vitamins to be fully functional. The sixth one is regulation. Enzyme activity can be regulated by various mechanisms to ensure that reactions occur at appropriate rates. These mechanisms include allosteric regulation, which is binding of regulatory molecules at sites other than the active site, which changes the enzyme's shape and activity. Feedback inhibition. The end product of a metabolic pathway inhibits an enzyme involved earlier in the pathway. Post-translational modifications. Chemical modifications such as phosphorylation can activate or deactivate enzymes. Properties of an enzyme. Enzymes are complex macromolecules with high molecular weight. They help in the breakdown of a large molecules into small molecules or bring together two smaller molecules to form a larger molecule. Enzymes do not start a reaction. However, they help in accelerating it. Enzymes affect the rate of biochemical reaction and not the direction. Enzymes are specific in action. Enzymatic activity decreases with increase in temperature. They show maximum activity and an optimal pH of 6 to 8. The velocity of enzyme increases with an increase in substrate concentration and then ultimately reaches maximum velocity. So classification of enzymes. Enzymes are classified based on the type of reaction they catalyze. The Enzyme Commission EC has developed a numerical classification system that categorizes enzymes into 6 major classes, each with subclasses and specific enzymes. The first one is oxidoreductase. Function, it catalyzes oxidation reduction, that is redox reactions, where electrons are transferred between the molecules. Example, dehydrogenase, which is remove hydrogen atoms. Example, lactate dehydrogenase. Oxidase, transfer oxygen from molecular oxygen O2 to a substrate. Example, cytochrome C oxidase. Reductase, add hydrogen atoms. Peroxidase, use hydrogen peroxide H2O2 as the electron acceptor. Example, catalase. The next one is transferase. Transfer functional groups, example methyl, glycosyl, phosphoryl, from one molecule to another. Example kinases. Transfer phosphate group from ATP to substrates. Example hexokinase. Transaminase. Transfer amino groups between amino acids and ketoacids. Methyl transferase. Transfer methyl group. Example DNA methyltransferase. Hydrolase. Catalyze the hydrolysis of various bonds, that is CO bond, CN or CC bond, by adding water. Example proteasase. Hydrolysis peptide bond in proteins. Example trypsin pepsid. Lipase. Hydrolyze ester bonds in lipids. Example pancreatic lipase. Nuclease. Hydrolyze phosphodiester bonds in nucleic acid. Example DNase, RNase. Phosphatase. Remove phosphate group from molecules. Example alkaline phosphatase. Lysis. Catalyze the addition or removal of groups to form double bond or reverse or break the bonds without hydrolysis or oxidation. The first one is decarboxylase. Removes carboxyl groups. Example pyruvate decarboxylase. Aldolase. Form carbon-carbon bonds. Example aldolase in glycolysis. Synthesis. Catalyze the joining of two molecules without ATP. Isomers. Catalyze the rearrangement of atoms within a molecule, converting it from one isomer to another. First one is racemase. Convert optical isomers. Epimerase. Convert stereoisomers. Mutases. Transfer functional groups within a molecule. Ligases. Functional. Catalyze the joining of two molecules within the concatenate hydrolysis of a diphosphate bond in ATP or a similar triphosphate. Example are synthetases, carboxylases, DNA lysis. Enzymes are categorized based on the type of reaction they catalyze, which allows for a systematic understanding of their roles in biological processes. This classification aids in study and application of enzymes in various fields, including machine, biotechnological, and biochemistry. Functions. Enzymes help in signal transduction. The most common enzymes used in the process includes protein kinase that catalyzes the phosphorylation of proteins. They break down large molecules into smaller substances that can be easily absorbed by the body. They help in generating energy in the body. ATP synthesis is the enzyme involved in the synthesis of energy. Enzymes are responsible for the movement of ions across the plasma membrane. Enzymes perform a number of biochemical reactions including oxidation, reduction, hydrolysis, etc. to eliminate the non-nutritive substances from the body. They function to reorganize the internal structure of the cell to regulate cellular activities. Summary. Enzymes are the proteins that function as biological catalysts. Enzymes catalyze all aspects of cell metabolism. Enzymes are highly specific to the reactions they catalyze. They alter or speed up the rates of chemical reactions they occur in a cell. They remain unchanged after chemical reactions. They are affected by temperature and pH. They catalyze reversible reactions. Thank you for watching this video. Stay tuned for more such videos. Please do like, share and subscribe.